Today, we are diving deep into one of the biggest mysteries of all, heredity. We're going on a journey to crack the code of life itself, to figure out how it's all passed down and how it creates every single unique person and creature on this planet.
You know, it's a question we've all asked ourselves at some point, right? You look in the mirror and you see bits and pieces of your family looking back at you. But how does that actually happen? How are these traits dealt out generation after generation to create that one-of-a-kind mix that is, well, you? Our whole story kicks off in a pretty unlikely place, a quiet monastery garden. It's here that one man's incredible patience and observation would literally create the foundation for the entire field of genetics. And this is our guy, Gregor Mendel. Now, while everyone else was busy debating these huge, sweeping theories about life, Mendel was in his garden meticulously crossbreeding thousands and thousands of pea plants. He wasn't just looking, he was counting, he was calculating, he was searching for patterns and things like seed color and how tall the plants grew. And boy, did he find some patterns. His first massive discovery was what we call the law of dominance. He figured out that for any given trait, you get two factors, one from each parent. But if those factors are different, let's say one for purple flowers and one for white, only the dominant one actually shows up. So you see purple flowers. But, and this is the genius part, that white factor, it's not gone. It's just hiding, masked, waiting for its chance to be passed on. And this is where it gets really cool. When Mendel started tracking two different traits at the same time, like the shape and the color of the peas, he found this super consistent 9:3 to 3:1 ratio. Every single time. This wasn't just a coincidence, it was practically a mathematical law. And that predictable pattern was a giant clue that heredity wasn't some messy blending of traits, but a system with very clear, very predictable rules. This led him to his second big rule, the law of independent assortment. Basically, it means that the factor for pea shape has no influence on the factor for pea color when they get sorted into reproductive cells. They're totally independent. Think of it like shuffling two separate decks of cards and then dealing one from each. This is what makes all those different combinations in that 9:3:3:1 ratio possible. Okay, so Mendel's work was absolutely groundbreaking, but it left a huge question just hanging in the air. These factors of his were a brilliant idea, but what were they physically? And where in the world were they hiding? And for decades, nobody knew. It was a total mystery. Scientists could see cells under a microscope, but connecting Mendel's invisible rules to something they could actually see inside a cell, that was the next giant leap. And here come our next two detectives, Walter Sutton and Theodore Boveri. Right around the turn of the 20th century, they were studying how cells divide to make sperm and eggs, a process called meiosis. And they kept noticing these little thread-like things called chromosomes, and they were behaving in a way that seemed really, really familiar. And boom, here is the aha moment. Just look at this side by side. Mendel said his factors come in pairs, while chromosomes come in pairs. He said factors separate and get sorted independently. Chromosomes do the exact same thing during meiosis. The behavior wasn't just similar, it was a perfect one-to-one match. This incredible observation gave us the chromosomal theory of inheritance. All of a sudden, Mendel's abstract factors, which we now call genes, had a physical address. They were riding on the chromosomes. The mystery was solved. Or was it? Because in science, as soon as you think you have it all figured out, the plot thickens. The story is never quite that simple, and researchers started finding exceptions that made things even more fascinating. So it turns out that law of independent assortment wasn't always true. Scientists found that some genes didn't sort independently at all. In fact, they seemed to travel together as a package deal, almost like they were physically stuck together. And they were. This is called linkage, and it happens when genes are located close to each other on the very same chromosome. But here's another twist. Even these linked genes can be separated sometimes. There's this amazing process called crossing over. When the paired chromosomes line up before a cell divides, they can get tangled up and literally swap pieces of themselves. It's like cutting and pasting segments between them, which shuffles the genetic deck even more and creates brand new combinations of traits that didn't exist before. Now let's zoom in on one very special pair of chromosomes, the ones that determine our sex, because they have their own unique rules and carry some really interesting traits along for the ride. In humans, it all comes down to the X and the Y chromosomes. Females have two X's, so every egg they produce always carries an X. Males, on the other hand, have one X and one Y, so their sperm can carry either one. So you can see right here, it's the sperm that decides. If an X sperm wins the race, the baby is a female. If a Y sperm gets there, it's a male. And this has huge implications, especially for traits on the X chromosome, like red-green colorblindness. Take a look at this chart. A colorblind father can't actually pass the trait to his sons because he gives them his Y chromosome. Instead, he passes that colorblind gene on his X chromosome to all of his daughters. Now, they usually don't show the trait themselves, but they become carriers, which means they can then pass it on to their kids. And that unique inheritance pattern we just saw, it has a name, crisscross inheritance. It's like the trait crisscrosses from a grandfather through his daughter to his grandsons. It looks like it's skipping a generation. And when you see that pattern, it's a classic giveaway that you're looking at a trait linked to the X chromosome. You know, this whole intricate dance of chromosome splitting and sorting is usually perfect, but every once in a while, there's a misstep in the choreography. And that one mistake can have really profound consequences. And those missteps are what lead to chromosomal disorders. It's really an issue of numbers. They happen when a person ends up with too many or too few chromosomes, or maybe just a piece of one is missing or has been stuck on in the wrong spot. So, here are a few well-known examples. Down syndrome, which was first described way back in 1866, is caused by having an extra copy of chromosome number 21. Then there's Turner syndrome, which happens in females who only have a single X chromosome, that exogenotype, which leads to being sterile. And on the flip side, Klinefelter syndrome is when males have an extra X chromosome, making them XXY, which also results in sterility and more feminized characteristics. This right here, this is a karyotype. It's basically a photograph of a person's actual chromosomes, all lined up in order. And if you look really closely at the pair labeled number 21, you'll see there aren't two, there are three. That is the genetic signature of Down syndrome. It's a crystal clear picture of what one of these chromosomal disorders actually looks like. So why does any of this really matter? Well, think about it. From Mendel's quiet garden to our own DNA, understanding all this helps us make sense of our own families. It lets us identify carriers of genetic diseases, which is huge. It explains the incredible, beautiful diversity of life on Earth, and it's what has paved the way for mind-blowing technologies like genetic engineering. We have come so far from those humble pea plants. We figured out the fundamental rules of the game, and we've even started to read our own instruction manual. The journey has been absolutely incredible. And it just makes you wonder, what's the next big chapter in our genetic story going to be? What mysteries are we going to solve next?